Okay, so it's 9 o'clock. Hello, everyone. Welcome to our today's Immuseum seminar. Thanks everyone to join us during the weekend, and particular the big thank to our today's speaker to spend the night in China from Tsinghua University with us, share his study. So it's really my pleasure to welcome our today's speaker, Dr. Zhang Conggang from Tsinghua University. So Conggang currently is associate professor in School of Pharmaceutical Sciences in Tsinghua University. Conggang complete his undergraduate study in microbiology from Shandong University, and then actually he moved up to switch his study focus to structure biology, also in Shandong University, complete his master's degree. After this, Conggang moved to US to pursue his PhD training in biochemistry in University of Colorado Boulder, and then complete his training 2015. So after Conggang complete his PhD training, he decided to move to UT Texas Southwestern Medical Center to do his postdoc fellow training with James Chen, and then focus in studying biochemistry and structural biology of cGAS-STING pathway. So from 2019, Conggang moved back to China and then studied his independent research program at Tsinghua University School of Pharmaceutical Sciences. Last year, he was promoted to associate professor. So Conggang's research group has been engaged in basic research in this field of innate immunity and mitochondria quality regulation for a very long time, particularly focusing on the field of DNA innate immune signaling pathway, PINK1 or parking signaling pathway, autoimmune disease, bacterial, and inflammation. So his lab utilized a combination of cell biology, structural biology, biochemistry, and autoimmunology method to study the core mechanisms and potential applications in the field of anti-infections, anti-tumor immunotherapy, anti-inflammatory treatment, and neurodegeneration diseases. The current research work in his lab mainly focus on a few different aspects, including one, mitochondrial quality regulation and innate immunity regulation mechanism. Second is bacterial nucleotide metabolic pathways and immune regulation machinery. And then last part, he's studying the possible application of the above machinery and the process in the treatment of inflammation, autoimmune diseases, and neurodegenerative diseases. So I think without further ado, I think Conggang gonna to talk a bit of his recent work regarding to the cGAS-STING pathway. So really appreciate Conggang being with us today, and we are very much looking forward to your talk.
All right. Thank you. Thank you. Thanks to Trent's nice introduction and this opportunity to present here. Today, I will give an introduction of our work on the cGAS-STING pathway. So for today's talk, I will start from three different parts. The first part, I will give a quick introduction of the background of our working background of the cGAS-STING signaling pathway. And second, the third part is two group of studies. So about the STING trafficking and the cGAMP. So the second part about STING trafficking, and the last part is our work on cGAMP. So cGAMP is the second messenger of the cGAS-STING pathway. All right. So our work is working on innate immunity. So innate immunity is the first line of our immune system. So it when the pathogen is here, so the innate immunity will be activated very quickly at the early time points. It will work through some immune cells like macrophage, like DC and NK cells. So DNA could active in the immune response. So this is an important part of the immunity. So it has been known for a long time, like more than 1 year ago, that people know DNA can active in the immune response. So in 2008, the adapter protein of the DNA sensing, the STING protein is identified. Then the DNA sensor cGAS and the second messenger cGAMP is identified in 2012. So now people know the cGAS-STING signaling pathway is the major pathway for DNA sensing. So this is the model for the cGAS-STING pathway. So we could see DNA in the cell cell, which is to know the DNA could form the dead cell, could form a virus, could form like bacteria. The DNA could also be like it's released from damaged mitochondria or damaged nuclear. So when DNA is in the cell cell, it could be sensed by the DNA sensor cGAS binds STING. Then it will use ATP and the GTP as substrate to produce the second messenger cGAMP. cGAMP will bind the downstream adapter protein STING. Then they will active like STING TPK1 signaling pathway to induce type 1 interferon. So it also could active the mCARB signal response. So this is like the major model people know now about the cGAS-STING pathway. So this is a video that I took like 10 years ago. So it's a HeLa cell. So I stable experience STING. Then I use a method to deliver cGAMP into these cells. You can see STING, which is a ER protein. It will form puncta very quickly. You can see the puncta like in very clear, like in half an hour. So now we know it's a trafficking process. It's a STING from ER trafficking to Golgi, and then finally, like if you take the video for a longer time, it will come to a lasum. So it's a beautiful trafficking process. So this is the signaling transduction for this process. So 10 years ago, when I get this image, this looks very amazing. So that looks that so there's very clear you can see there's some very open questions. For example, so how this STING, this puncta is assembled. So what's the mechanism for this state trafficking process, like from ER to Golgi and to lysosome? So also how the downstream, like through here, cGAMP binds STING, how this STING TPK1 and ARP3 phosphorylation signaling cascade is activated, and how STING binds you. So there's a lot of questions here if you look at this video. So to answer this question, so the first key question is the structure of STING. So at this time, we use a PREM method and collaborate with the lab and the Zhang Xuewu lab. So we saw the structure of STING. This is the chicken STING. It's in its APO form and the cGAMP binding form. So this is the picture for the full length STING. So this is the transmembrane domain, and this is the cGAMP binding domain, and it have an N-terminal and the binding with the cytosol binding domain. And then it form a very beautiful, very tight structure. cGAMP is bound here. So what we see from this structure is when cGAMP binds STING, like you can see here. So this cytosol domain will have a rotation, like it's a 180-degree rotation. So around this linker region, so that it will be activated. So from information, so it's a very big change from the confirmation change if it's look at it from the APO form and the cGAMP binding form. So that's a STING related autoimmune disease. So it's called the name is SVI. So the mutation of STING. It's called the gain of function mutation. So it's an autoactive of STING. For example, this mutation V155M mutation. So it causes inflammatory response, and the people will have some immune response in different tissues. So if we look at the structure, we can find the potential mechanism for this autoimmune disease, because this mutation is happened to this linker region. So if we believe that we hypothesis if this V is a smaller amino acid, if it's mutated to a bigger amino acid, so this linker will be forced to have a 100-degree rotation. So STING will be autoactivated. So this could explain why this mutation could cause this gain function mutation, and this activation is independent of the cGAMP binding. So then another important information we get is from the cryo-EM studies. We get the tetramer structure of STING. So we can see from structure. So before cGAMP binding, before the rotation of STING, so there's a region that is that auto inhibition. That's the confirmation. If the cGAMP bind, this region will come a little confirmation change. It will be decreasing and the form and this two STING dimer will become from auto inhibition state to auto like a binding state. So that's the reason why we have cGAMP here, cGAMP binding to the SARS domain, and then STING will have a 180-degree rotation, and this structure will be a little closed, and then this STING dimer will change the form auto inhibition to a oligomerize conformation change. So we will see a STING oligomer. So this study, based on this study, we continue to look at the STING and the TBK1 complex. We try to get the STING and the TBK1 complex structure. So this is the structure we finally get. So finally we get this is a STING dimer binding to a TBK1 dimer. And STING binding the TBK1 using is a C-terminal that is very flexible tail and form this very dynamic complex. Actually, this complex, the resolution is not so high. It's just but we can see this is a complex. So the information we can see clearly is we can see the interface, because if we look at the TBK1, we will find like if it's a normal TBK1, there's no this information here. If you have STING and TBK1 together, you will see this very clear and very high resolution TBK1 binding here. And because TBK1 tail is very flexible, so you cannot see a structure from both like a crystal study or the people from a cryo-EM study. You cannot see this information. But if you mix them together, you will see the binding interface. So this I say is a very high resolution information. We could see clearly TBK1 and the STING binding motif. So this is a binding motif. So STING use this motif binding with TBK1. As this surface is formed by two TBK1, and this is the STING C tail. And this side is the phosphorylation side of STING by TBK1. So then we verify that this interaction is very important. So we do mutation to this interface, like we do mutation to STING. You can see if you mutate any residue that in this interface, you will get decrease the TBK1 STING-R3 phosphorylation signaling. You will if you do mutation, this STING and the TBK1 interaction in cell will disappear, and also the type 1 interferon response if you do mutation. So you will just get impaired like a type 1 interferon response. So another important information we see from the structure is the S366 phosphorylation side of TBK1, the STING by TBK1 is very far from the TBK1 kinase active side. So we'll say it's like is a 30 angstrom. So this means it's impossible that TBK1 to phosphorylate the STING S366 residue that is bound to. So this could explain STING must form an oligomer. So in this oligomer platform, so TBK1 will phosphorate the other STING CTL because it cannot phosphorate the S residue that it bind to. It only can phosphorate the tail that is close to it. So in this oligomer platform, TBK1 will first auto-active and then will phosphorate the other like tail that around it. So this is the total model for this two-story. So like cGAMP bind STING will have a 180-degree rotation and will form oligomer. So in this oligomer, STING will use a CTL bind with TBK1. And in this platform, TBK1 will first will be auto-phosphorate by the other TBK1 kinase, and then active TBK1 will phosphorate the STING CTL that is close to it, but not the tail that bind to it. When TBK1 is phosphorated, it will recruit ARP3. So ARP3 will then phosphorate by TBK1. Then the phosphorated ARP3 will form dimer, then it will translocate to a nuclear and then active the internal immune response. Okay, so this is the working background of the cGAS-STING pathway for our work. So for the next part, I will talk about the interaction of STING with other signaling pathway for the STING trafficking. So this work is started from the MCAB in the immunity. We know MCAB signaling is important for innate immunity. So in this picture, we can see like MCAB is closely related to many inflammatory diseases. Actually, MCAB could be activated by many like ligands. For example, it could be activated by toll-like receptors. It could also be activated by R1 beta and TNF alpha and the EGF. So there's many signaling pathways can activate MCAB signaling. So this show the toll-like receptor signaling pathway. So like many material can activate MCAB signaling. For example, the LPS toll-like receptor 4 signaling pathway is very famous. Like other components from bacteria could also like activate toll-like 1, 2, and also there's a toll-like receptor 3, also like 4 and 7, 8, 9 is in the endosome to activate in the immune response. So all these toll-like receptor pathway could activate a very robust MCAB signaling. So we found that all the toll-like receptor agonists could enhance the STING type 1 interferon response. So this is a several example. For example, we use a LPS, which is a toll-8, a toll-4 and R848, which is toll-like 7 and 8 agonist, and also HKLM, which is a toll-2 agonist. So we can say like in normal cell, like the type 1 for the type 1 response in T1 cell, cGAMP can like active like a 10-fold, like or 20-fold immune response. But if you add LPS or R848 or HKLM, you will see a very robust synergistic effect for the type 1 interferon response, because this toll-like receptor do have a very weak activation to the type 1 interferon. So, but you mix them together, you will see like you can see like sometimes use HKRM, you can see like more than 200-fold response. It works in the to the cGAMP and to the other STING ligands, like 3,3 cGAMP, CDI-AMP, CDI-GMP, and like is other small molecular compound that active STING. So then we look at the more MCAB signaling. So besides the toll-like receptor signal, we found that like R1 beta and the TNF alpha and the PMA and the EGF, we all could have some synergistic effect to activate the STING top one interference signaling. So then we look at the mechanism for what is a synthetic enhancement effect. So we found this is STING dependent. This is no surprise because it's enhanced. It's happening in STING. So the surprising thing is we found finally we found it's dependent on P65. So this is a little surprise because the P65 is very downstream of the MKB classical signaling pathway. So we test many components like a TBK1 and also a lot of upstream, but it didn't work. So we finally found it's a P65. So that means there should be a mediate between the P65 and the STING. So that is responsible and regulate this enhancement effect. So then we do a lot of experiments trying to find out which is the mediator that regulates this enhancement effect. Finally found is this classical MKB signaling activation where the mitochondria destabilize. Like we use R1 beta as a sample because R1 beta could active a very strong MKB signaling response. We can see like in wild-type cell, if we add R1 beta, we can see this mitochondria become destabilized. But if we knock out the P65, this destabilizing effect is disappeared. But as control, if we use the inhibitor of mitochondria to inhibit this mitochondria assembly, so it's not dependent on P65 at all. So then we looked at why this microtubule destabilization could cause the STING enhancement. So finally, we found that is this microtubule destabilization by MKB activation could change the SNIR trafficking process. So I'll talk about it later why this microtubule network is very important for SNIR trafficking. So today I just show the conclusion. So for the wild-type cell, if you use R1 beta to treat the cell with cGAMP, you will see STING will become a puncta. With cGAMP, you will see a puncta around the perinuclear region, just as what I show in the video. So you will see like it become a puncta. But with activation by R1 beta, it cannot form puncta. If you knock out the P65, so both of these two groups is puncta. So here you will get a more STING signaling around the cells. But if without this signal, it's a normal ER to Golgi become puncta and finally become degraded. So for this work, first I will give a model for this story. We found that all classical MKB signaling activation, including polar receptor on beta and the TNF and the other signalings, if you active MKB signaling, you will get mitochondrial microtubule destabilized. So in this mitochondrial destabilized cells, the STING trafficking process will be changed. So the STING, without this pathway, STING with cGAMP binding will traffic from ER to Golgi. But with this signaling, it will become another signaling pathway. Seems the STING signal will become dispersed in the cell and will not become condensed puncta. So besides this MKB work, we also do a lot of drug screening. We try to find like a compound that will regulate the cGAMP STING signaling. We try to find the enhancer or inhibitor of this signaling pathway. So we do a lot of screening. We found a compound is called pedophilics. So HFD drug that used to treat for HPV virus for like many years. So it's a classical microtubule destabilizer. So we find that this compound also enhances the STING signaling. So this compound itself cannot activate the top one inhibitor. But if we will use together with cGAMP, like cGAMP is here, we add pedophilics, we will see a enhanced effect of this STING signaling. And this enhancement, you could see it like even in like 1 nanomolar. So even in sub one below than 1 nanomolar, you still see the enhancement effect. then we find that this enhancement is STING dependent. So this is no surprise, and it's also from the phosphorylation signaling from the inference expression, and also from the RSRE reporter assay. We also found that the enhancement only happened to cGAMP-STING signaling. If we use sending virus to active like the mouse RNA mouse signaling, or use LPS to active the TORLAC4 receptor, so there's no any enhancement. So the enhancement is specific to STING signaling. So then we look at this STING GFP puncta. So this is a HeLa cell with a STING GFP stable expression cell. We can see cGAMP treatment, you will see a puncta in the perinuclear region. If you add pedophilics to cGAMP, so it's changed, everything is changed. It's completely changed. So the result is very black and white. So you can choose, you can take a big picture, like 100 cells. You will also get like a 100 percent black and white result. They are completely different. If you look at the image, you can see that there's another compound, the first compound that active the STING. There's another mechanism. Maybe I can talk about it in the next talk. So there's a video we take. So this is the control STING GFP cell. This is pedophilics itself. No effect to the STING signal. If you add cGAMP, so this is a very clear perinuclear puncta. If you add cGAMP and this mitochondrial destabilizer, you will see it cannot become a puncta. So we do a lot of stringing. Actually, we besides this pedophilics, we search out many other compounds. So all the mitochondrial destabilizer are identified, like in the nukudazole, like MAE, like this is another compound. So all these compounds several of these compounds are FDA pro drugs. So we string all them out. So it means this mitochondrial destabilizer reagent could enhance this STING signaling. So we also do a research in antivirus experiments. We choose MMAE as an example because MMAE is the first it's the first compound that used in ADC. So it's also imported by FDA for drug treatment clinically. So we use this compound together with cGAMP. We can see a very robust antivirus response in mouse and also in cells. So I think this is because two reasons. First reason is because virus need this microtubule network somewhere, like a DNA virus need this microtubule network to a transmission and to a transport. If we impair this microtubule network, we will impair the virus transfection. I think, but maybe the more important reason is because the enhanced effect of this combining strategy with the STING signaling. If you use cGAMP and MAE together, you will get enhanced STING signaling, and you will get also very robust type 1 interferon response. So you will get a very robust antiviral effect, both the pre-treatment and to the treatment during the virus infection. So together for this part, we published 3 papers. One is about the MCAR-B signaling and the STING signaling synergistic enhancement to the STING signaling, the type 1 interferon response. The second 2 paper is about 2 microtubule destabilizer. So these 3 stories are targeting to the same mechanism, but the STING is targeting the STING trafficking mechanism. So I mentioned the STING, once it binds to cGAMP, it needs to traffic from ER to Golgi and then finally to lysosome. So in the Golgi, they'll be activated to active like type 1 interferon and also MCAR-B, also autophagy response, and then in lysosome, they'll be degraded. So if you use MCAR-B activation or the microtubule destabilizer to change this STING trafficking process, we can increase the STING signal at Golgi, and at the same time, we can increase this STING degradation process. So I think this is the reason why this strategy could cause the sting signaling enhanced. Okay, so this is the first two part. I give an introduction of the cGAS-STING molecular mechanism, and also our several studies about sting trafficking mechanism. So I use like 10 minutes maybe to talk the last part about 2 hour studies on cGAMP. So the second messenger of the cGAS-STING pathway. So I mentioned like this is a model for the cGAS-STING pathway. So cGAS bond DNA use ATP and GTP to produce cGAMP. cGAMP is here. A negative charge hydrophilic compound. So it's about 7 Dalton. So 700 Dalton. So it's negative charge and hydrophilic. So there's a several questions about this open question about this second messenger. So the first question is how it could cross the cell membrane. So the second question is how it's degraded. So the stories start from our work on the antibacterial peptide. So we are working on the bacteria and the host interaction. So one of our postdoc is working on antibacterial peptide. So because antibacterial peptide is also an important part of the innate immune system. So like for the many like insects, like fish, like frog, so they are important for their immune system. So when we are learning the bacteria with the host interaction, we found that an important antibacterial peptide LL37 could have an enhancement effect with cGAMP. So cGAMP is here. If you like 1 micromolar or 2 micromolar, you can see a very weak immune response. But we add LL37, so you will get a very robust immune response. So it's a more than 100-fold change. And this response, if you look at it from the Western blot, from the phosphorylation signaling, is dose dependent on the LL37. So then, because the cGAMP is from bacteria, we know that bacteria can produce like cGAMP, cGAMP, and cGAMP. We are more interested to cGAMP. So cGAMP is actually is produced from mammalian cells, also our cells. So we ask whether this LL37 could also active like cGAMP to activate this sting signaling. So indeed, the answer is yes. So we found that this is the response for cGAMP, like use 1 micromolar or 100 nanomolar that in the physiological condition. So the response is very low. If you use LL37, you will get a very robust enhancement effect. So the synergy is very clear, and it's also dependent. It could happen like in TH1C cell and raw cell and BB2 cell. So this is a classical like immune cells we use in the labs. You can see the clear LL37 activation to the cGAMP signal in these cell lines. So because time reason, I'll just give a summary for this story. So finally, we found that LL37 is one of the most important antibacterial peptides in our immune system. Will bind cGAMP and deliver it between cells to enhance the immune response between cells. So this is a current overview of the cGAMP transport system. It's a review paper written by Dr. Xie Wei and Ding Shou. So they show the cGAMP transport system. Like cGAMP could be transported by a virus. Like if a virus infects one cell, the cGAMP produces the combined to the virus particle and then transport to other cell and also to amplify the immune response. cGAMP also can be transported by different transporters. Like here, people show like cGAMP also can be transported by LL37. So if you look at the immune response, so LL37 driven this immune response is the most robust in all these systems. So it can really robustly active this STING signal. So this is the last story. So we also are working on drug screening. So we are trying to looking for cGAMP STING signaling inhibitor. So for this screening, we finally found a compound. We call it, the name is T009. We found that this compound could selectively inhibit the cGAMP-induced immune response, but not inhibit like other activator of the STING signaling. Like here, we show this SR717 is a small molecular compound STING agonist. So this T09 can only inhibit cGAMP triggered STING signaling, but not SR717 induced immune response. So based on this information, we have a hypothesis. We think maybe this T0009 could induce an enzyme. So this enzyme selectively degrade the cGAMP. So this we think this maybe this is the reason why this compound can only inhibit the cGAMP-induced immune response, but not other compound induced. Then we try to get this enzyme. So we get a lot of TGP1 cells. We use this E09 compound, and then we make a bead that bind to a cGAMP analog. This analog could resistant to degradation. Then we do PODOL assay to try to find this enzyme. Finally, we get this enzyme. The enzyme is SMPDL3A. Indeed, this we purify this enzyme. It can clearly do a cleavage to cGAMP. So this is ion column assay. We can see the cGAMP is cleaved. This is the TLC. You can see this is the cGAMP. You add this enzyme, it will be cleaved. And we look at the function assay. We can see this enzyme will selectively degrade cGAMP, but not 3,3 cGAMP and cDAMP and cDGMP. So it selectively degraded this cGAMP. So then we do some biochemistry assay to verify this model. So this is the structure study. So we saw the structure of this SMPDL3 binding with a cGAMP analog. So we get the structure, and we finally solve the structure. We finally get is a dimer conformation. So this is the first time people see this dimer conformation. So we can see if without cGAMP or cGAMP analog, you cannot see this dimer conformation. So there's several structure solved for this 3A structure, like 3A binding to other substrate. It's monomer. So now its Apo form is also monomer. So it's with cGAMP analog, we capture this dimer information. So we think maybe this dimer information is important for its enzyme activity. We do mutation around this dimer interface. You can see here, if you mutate this dimer interface, so the cGAMP cleavage activity is almost gone. So this dimer, this transient dimer, the dimer information is important for its activity. So we also verify that use cellular assay and use mouse assay. So this is an example for a cellular assay within parm cell. If you knock out 3 protein, you can see in wild type cell, T09 will have an inhibition effect to the cGAMP-induced STING signaling. You will see a decreased phosphorus STING signaling. But if you knock out 3, so this inhibition effect is completely disappeared. You will see an enhanced expression of the phosphorylation of STING signaling. So LSR transcription factor is the upstream of this 3 protein. We also use in IBM cell, we knock out this LSR transcription factor. You can see if you knock out LSR, the 3 expression is decreased, and the T09 induced the inhibition effect is also decreased. So this is the working model. So basically, we find the T09 is a specific inhibitor of the cGAMP-STING signaling. And this 3 protein, SMPDR3 protein, is a new enzyme for the cGAMP degradation. So the working model is the T09 will induce the LSR lipid metabolism activation. So the LSR transcription factor will induce the expression of 3 protein, and the 3 protein will be induced by cGAMP to form a dimer, and this dimer will do cleavage to cGAMP, and finally get to shut down this signaling. So this signaling, the LSR signaling pathway have been known for like more than 20 years. So they have two group of functions. First is the regulation of lipid metabolism. So the second function is anti-inflammatory response. So I think our work provides a mechanism for why this LSR lipid metabolism activation could anti-inflammation through like inhibit cGAMP-STING signaling pathway. So because several LSR lipid metabolism agonists is on the clinical trial, so I think people maybe can switch some of this trial to a anti-inflammatory response, like related to the cGAMP-STING signaling, to see if it works in the anti-inflammation. So this is a cranial model that people summarize in immunity about the cGAMP signaling and the regulation network. So cGAMP could be transported by different transporters, and it also can be degraded by different enzymes. So this is SMPDR3A is one of the enzyme, and the EMP1 and the EMP3 is other enzyme can degrade the cGAMP to control the homeostasis of cGAMP level in cells. This is my last slide. So we can see that in the last like 10 years, the cGAS-STING signaling pathway is expanding. So we see a lot of crosstalk of the cGAS-STING signaling pathway with other pathways. So today I talked about the two group story. It's a little related to the infection, the other in the immune response, because I talked about mCARB signaling. Also a little about inflammation and the metabolism, lipid metabolism, and also a little about tumor. So I believe that there'll be more people that can get more and more these exciting discoveries to this field. So finally, to the funding support our work, the lab to working hard to do this project. And this is Zhimeng, Lulu, and Hanjing. They are performing the project that are presented today. Finally, thank you for your attention. So I'll be happy to take any questions.